Okay, there we're going. So thank you to everybody that have joined us today. We are very happy to have another webinar, and that Kelsey could get as a speaker, and she will tell us more about it. And as Steve just said, he's seen the writing on the wall, and machine learning is something that is going to be part of our lives in the future. And I think that's why there's also a lot of excitement about this talk today and how we can apply it in different fields of taxonomy. Our society is really consisting of botanists, zoologists, mycologists, bacteriologists. So there's quite a range, and it's always nice for us to see what people have done in other fields, so that we can also think about it and apply it. So with that, I'm giving over to Kelsey to introduce our speaker.
Thanks, Venice. So today I'd like to introduce you to Professor Alex Pyron. I met Alex back when I was in grad school, and Alex was interviewing for a job there, and we're very glad that he got it. So Alex is the Robert F. Griggs Associate Professor of Biology at George Washington University in Washington, D.C., but he's also a research associate at the Division of Amphibians and Reptiles at the Smithsonian in D.C. as well. So Alex did his grad school at CUNY, or the City University of New York, with Frank Burbrink, and then a postdoc with John Weens following that. His lab studies diversification and speciation of global biodiversity, and does this from a herpetological perspective, although he does have interest in paleo and other groups as well. So thanks very much, Alex, for joining us today and telling us about machine learning and species delimitation.
Well, thanks for having me. So I put this talk together to explain a little bit of the research I've been doing recently using machine learning algorithms and some of the techniques that we can get from artificial intelligence and machine learning for data analysis, and how that can sort of complement a lot of the questions that we traditionally or historically have been asking in systematics. And so a lot of the questions in systematics or taxonomy are still the same. We haven't changed what we want to know about discovering and describing and delimiting new species, but the way that we do that is shifting dramatically. And I think that a lot of these techniques are going to be helpful. So I have this divided into three major sections, each of which is covering sort of a really distinct topic, and I'll just give sort of a high-level overview, but I think we'll see how that they can tie together in a way that will potentially be really helpful for the field of systematics. So first, I'll talk a little bit about machine learning and statistics and how we actually use and analyze data and how that can change. Then we'll talk a little bit about the species problem, something I imagine most of you have thought a great deal about. How do we define taxa? What is it that we're trying to actually analyze? And then sort of bringing those together, what can we think about in terms of integrative taxonomy, not only how we study species or speciation, but how that ties back into our broader understanding of evolution as well. So first, let me just give you a little bit of background on machine learning. And so obviously this is a huge topic. I'm sure you read stories about ChatGPT and things like that every day. And there are aspects of even the stuff that I'm talking about now, they're actually shared with even large language models and some of the generative AI that you hear about in the news. And so the field is very broad, but a lot of the techniques and algorithms are actually shared among a lot of those applications. So I'll start with a really famous paper from 2001 by one of the earliest figures in machine learning, Leo Breiman. And it's kind of amazing. He was born, I think, in the 20s. He published this paper when he was in his late 70s, and he was still generating, new algorithms for machine learning. And so it's kind of amazing that he was making those massive contributions that late in his career. So perhaps there's hope for all of us. But he gave this lecture, and then he turned it into a paper about the two cultures. And so what he identified was that there were two major approaches to data analysis. He said one assumes that the data are generated by a given stochastic model, and this is sort of classical statistics. And the other uses algorithmic models and treats the data mechanism as unknown. So in statistics, and I'll talk a little bit more about this, we think that the data generating process in nature is something real that we can learn about. like whether that's the mutation process in evolution or, the ecological processes that constrain population size, and we want to learn about those. But in certain instances, the complexity of the data generating process might be beyond our ability to infer at that level, and so we can sort of approximate it using algorithms, and that's machine learning. And so he says that the statistical community has been committed almost entirely to data models. And he took it even a step further. I'm not going to take it this far. He even went so far as to say this commitment has led to irrelevant theory, questionable conclusions, and kept us from actually working on really interesting problems. I think that might be an overstatement, but I agree with him, and I think most people agree with him when he says that if our goal is to use data to solve problems, then we need to move to a broader range of tools or a diverse set of tools that can allow us to answer questions from different perspectives. And so, for instance, in statistics or in data analysis in general, we typically have two major goals. One is to be able to either predict what the responses might be in the future based on input variables, or to extract information about how the inputs are linked to the outputs, learning about the process itself in nature. And in statistical analyses and statistical models, we're trying to learn about the data generating process by fitting a statistical model where the parameters of that model are actually the quantities in nature that we're interested in learning about. So even if we just have a basic linear regression, something like height in inches as a predictor, or height in centimeters, please excuse me, and then body weight in kilograms, the statistical model we fit, a best fit regression line, the parameters of that line, the slope and the intercept, contain information about the natural process. So this actually tells us something about the data generating process in nature. In contrast, if we're taking a machine learning approach, we might be interested more in a predictive function. And so, something like ChatGPT, what it's doing is it's taking your query and it's predicting what the next output in written English or whatever language should actually be based on its understanding of how words fit together. This is an algorithmic model that we fit. And the model itself sort of cancels out the need to understand, or not the need, but the desire to understand the data generating process. We're saying that what's happening in nature is potentially too complex for us to understand, but that we can approximate it with our model and make reasonable inferences about the world. And so two examples of this are algorithms like random forests and neural networks. And I'll talk a little bit about neural networks. But something like ChatGPT, or a large language model, is a neural network like this architecture on the right. And so one of the major early and still ongoing, reluctances or anxieties that a lot of people have about machine learning algorithms is that they often seem like they're leading us towards a lot of the basic statistical sins that we had drilled into us not to do. So things like data dredging, looking through thousands and thousands of variables until you find significant correlations, and then the false positives that can lead from that, where you can find all sorts of spurious associations between variables that may not actually be part of any real process in nature. And the way we try to avoid this in machine learning is through the use of what we call training data. So with training data, these are the input data that we use to build our model. And by training on known data, we can hopefully avoid overfitting the models so that we limit false positives and limit the effects of data dredging. And since we're biologists and we have training in statistics, we have training in the scientific method, what I want to kind of get at today, especially in systematics and taxonomy, is that we can combine both machine learning and statistical models, given our domain expertise, our subject-specific knowledge, to use things like machine learning to find potential mechanisms in nature that we can then test under a more rigorous or classical hypothesis testing framework with statistical models that will sort of give us a yin and a yang. We can data dredge to find potentially significant patterns, and then we can actually test those in a more rigorous framework to eliminate the false positives. The example that I always give my statistics classes or my biometry classes is, say we went back to the 50s before it was really established about the link between smoking cigarettes and lung cancer, and say a magic genie gives us a gigantic data set of, say, all of the people in the world and all of their lifestyle attributes and all of their health outcomes. And they give us a modern computer and some machine learning algorithms. Now, if you have, thousands and thousands of variables and millions of data points, and you just start going through and testing every single combination, classic data dredging, you're going to come up with a lot of false positives. So you might find that, say, lung cancer is associated with a lot of things, like listening to Perry Como, smoking cigarettes, or watching television. And so that's sort of our classic example of data dredging and false positives. However, since we are biologists and we have the scientific method, we can take these three positive relationships and then engage in rigorous hypothesis testing, creating crucial experiments, looking for causal mechanisms that might actually link these to the outcome of cancer instance, in which case we're going to eliminate TV and listening to the radio, and we're actually going to develop a biological or a medical model for the actual link, the true link between smoking and cancer. And so that's sort of my example of how we can, on the one hand, engage in these, behaviors we're not supposed to do, like data dredging, but through careful fitting and then through the application of the scientific method, sort of limit the negative aspects of that. So I'm not going to go through this whole graph here, but this is what we would call a simple neural network. And you can see how, extremely simple it is. There's only like 56 parameters here. But what I want to talk about is the idea of training. So just to give you the broadest possible overview, if we have a training data set where we know the inputs and the outputs, we can put the inputs in here on the left. The model will generate predictions, but we also already know the true values. So again, this is labeled training data. We can compare the predictions to the true values. And also, can you guys see my cursor?
Yep. We can see
Okay.
it.
So we can compare the predictions to the true values, and then we can optimize the parameter values to basically train the model to give us the most accurate possible outcomes. Every single line that you see here is a parameter. So, a linear regression model only has two parameters, the slope and the intercept. This is a neural network that only has two inputs and one output, but it still has something like, 30 or 40 parameters. So there's a huge amount of parameterization in these models. And when you hear on the news people talking about like the next generation of ChatGPT is going to have 150 billion parameters or something like that, this is what they're talking about. Each of these lines is a parameter. And we optimize those just using massive amounts of computing power. But ultimately, these models can oftentimes give us predictions or estimates that are very close to the true values. A classic example is just recognizing handwritten digits. So we have a 3 here, but we break that 3 down into these grid cells. You can see where the ink occurs or does not occur. It gets linked to all of these input neurons that are all linked together with weights and biases. I'm not going to go into all of that, but basically what happens is that due to the connections, the weights that have been trained for all of these neurons in this neural network, it ends up lighting up the output neuron of 3. So the algorithm can recognize handwritten digits and identify them with a very high degree of accuracy simply based on the connections between these neurons. And the neurons are very analogous to the neurons in your brain. They turn on and off. They're connected to each other. And so those connections and those activations give us sort of the underlying structure of how these models work. I know that's a really abstract introduction. It would, I could spend an hour talking about exactly how neural networks work. But I think for the purposes of this talk, just think about how we have input data. We have a black box, and it's fair to think about this as a black box, but the black box usually gives us very accurate answers. And so the question is, how can we use that to get at some of the fundamental questions in something like species delimitation? So in systematics, I think where this could be very helpful, and there's been an increasing amount of literature published on this in the last couple of years, is that it will allow us to incorporate big data sets, potentially with thousands or even millions of observations, because they're computationally very tractable. We can use data from multiple sources, not just things like DNA and morphological traits that we've traditionally used, but then potentially integrating other sources like climatic data, spatial data, things like that. And we can imagine, this is another neural network that I'll talk a little bit about. The blue input might be the genetic data, the red input might be phenotypic or morphological data, and the green input might be climatic or spatial data. And all of these can be integrated into a signal analysis to say delimit species or evaluate, conservation concerns over landscapes, things like that. And so again, we can build these really complex models and come up with hypotheses about, say, the existence of species, which we can then test using classical methods. I mean, if we have a hypothetical set of species, we could then go back and do old school breeding experiments. Can they actually interbreed with each other? Do they represent biological species? And so we sort of have the continuum of, extremely complex and abstract quantitative methods, but then we can always fall back on the fundamental biological mechanisms that we know to be important for evolution and ecology. And so with just that brief introduction to the sort of methods that we're using here, now I'll shift a little bit to something that most of you have probably thought quite a bit about, the idea of what we're actually trying to get at when we define taxa. So when I'm thinking about the species problem, I think less these days about species concepts, which tend to be in a more operational context. What explicit criteria are we using to define and delimit species? What I want us to think more about is the idea of species ontology. So what is a species in general, or what does a species represent? So ontology is the idea of thinking about existence, or thinking about what being means, whereas empiricism is what is and what we can measure. And so historically, species were often thought of as being classes or kinds, meaning that they have attributes, and if you have this attribute, you belong to this species. We are defined by those attributes. That has fallen out of favor, at least among a broader swath of systematists with a more philosophical bent recently, who now think of species more as being individuals. So obviously, individual organisms are individuals, but the species or the lineage that they belong to is itself some sort of individual that exists in space and time. So we could think about the phylogeny in this context, where we have individuals, we have populations, we have lineages. Those lineages are connected evolutionarily in the phylogeny. And each of these branches in the phylogeny represents some kind of evolutionary individual. You could think of it as a spatio-temporal entity. It exists in a given range of time and in a given range of space. And it has boundaries that are fuzzy while it exists, because some species might still be hybridizing with other species. And it has boundaries that are fuzzy when it begins and ends. There's no exact dividing line of exactly when speciation has occurred and exactly, well, there can be for extinction. That's obviously a hard dividing line, but that's pretty much the only hard dividing line. So the rest of the boundaries can be fuzzy, but for any species that we describe or delimit, whether it's Homo sapiens or Tyrannosaurus rex, that is a very discrete and distinct thing that exists in a certain range of areas through a certain range of time periods. In the case of T-Rex, it obviously went extinct 65 million years ago. And then with humans, it remains to be seen what will happen. But we all, as humans, share a common historical origin and a common set of phylogenetic, attributes that define us as this individual entity. That's not to say that any particular phylogenetic species concept is the correct, quote unquote, correct species concept. But what I want to kind of get at is, at least from the perspective that I'm taking, a species is defined phylogenetically, regardless of what criteria we actually use to describe or delimit them. And so if you are an individual and you are phylogenetically defined, you sort of exist regardless of the criteria. The criteria allow us to recognize species in nature empirically, but they don't define what those species are. And so this is just one philosophical perspective, and I mean, you're free to disagree with me, and there may be different opinions in the audience that we can talk about, but this is the perspective that I'm taking for the purposes of this talk. And so what that means is that our species problem then is defined less by these criteria. And so I'm going to pick on the biological species concept a little bit. And many of you are probably already familiar with the idea that the BSC can be problematic, for species that don't reproduce sexually, for instance. If the BSC is talking about actual or potential reproductive isolation or hybridization, it doesn't work well for asexual species. But there's another aspect in which it doesn't work well that I want to kind of go over here. with regard to actual or potential reproductive isolation, which is that any species concept, not just the BSC, but really any species concept that has a criterion for delimiting species implies the history of those species. If we define species as lineages that or populations that aren't interbreeding, and we have populations A, B, and C, if A stopped interbreeding with B and C first, and then B and C stopped interbreeding, that automatically actually implies the history of their speciation process, and the criterion of delimitation equals the tree. And it's actually almost always wrong. This is a really key insight from Velasco in 2008. This paper, really blew my mind, and I would encourage everyone to look it up, or at least nearly so. So it's not always wrong. But what I mean to say is that this might be the true gene tree that I just described. Population A branches off first, but it's still exchanging genes occasionally or hybridizing with this ancestral lineage here. And then B and C get split by a barrier, like a mountain range or a river. And so B and C don't exchange genes. They don't hybridize at all. But if A and B are still parapatric, they might still be interbreeding peripherally on their edges where they come into contact. What that implies is a species tree that is wrong. It's the opposite of the actual true evolutionary history of the group. It implies that A and B are more closely related because they were interbreeding more recently than B and C, which stopped interbreeding immediately once the barrier arose. And so again, this just sort of reinforces what I was saying before, that we have to think of species being defined phylogenetically as individual lineages that exist, spatiotemporally. If we define species purely based on criteria, these criteria are oftentimes going to imply histories. They're going to imply trees or imply phylogenies that will almost always be incorrect with respect to the true evolutionary history if we had full knowledge of that. And so what this leads to is a desire for taxonomy, for species delimitation, systematics, where we want to be able to discover species through things like collections and natural history, museums, things like that, or field work. We want to be able to delimit the species, through some sort of algorithms or quantitative models, whether it's machine learning or statistics. We want to be able to define them based on characters because we need, some objective reference for what we can actually use to recognize these species in nature. And we want to describe them taxonomically. For me, that's one of the most fun things to do, even though it's not the thing that, you get grant money or get big news articles or get promotions for. But, I've described like 50 or 60 species, and I think that's what a lot of us are initially, at least, really interested in. And it's something that still is probably one of the most long-lasting benefits that we actually produce, even after most of our other science has been forgotten, our species descriptions will still be there. And so we have all of these different potential ways of addressing this. And so what I'll talk a little bit about now is how machine learning might help us to tie them all together in a more cohesive framework. So if we start putting all of these different aspects of data and different aspects of inquiry together, how can we build an integrative taxonomy to study the evolutionary process in more detail? And for those of you who are familiar with the species delimitation literature, you know that the methods for actually just trying to define and diagnose species from data sets can vary a lot. You have dozens of algorithms. They almost all use genetic data. And so this is a classic paper from 2013 looking at skinks in Madagascar. And they were pointing out that each column here on the right is a different species delimitation method that they used, different computer algorithm, all based on the same data set, that gave massive variation in how many species each program said there was. So some algorithms might have said there's 9 species, some might have said there's 23 species. But almost all of these are also based on genetic data, mitochondrial data, SNPs taken from the nuclear genome. Very, very few algorithms have gone beyond DNA to incorporate things like trait data. There is an algorithm called IBPP that did a really great job of building a classical statistical model that linked the mutation process in DNA to the morphological, evolutionary process in traits. But the reason that that was very difficult is that, a classical statistical model has to have parameters that tie together what we know about the data generating process. But if we say we want a model that links together climatic evolution and phenotypic evolution and behavioral evolution and molecular evolution, it's easy to say that, but it's very difficult to implement because, you know, what is that process? What processes tie those together? What parameters do they have? How do we put that in a statistical model? How do we implement that in a computer algorithm? It may be difficult or impossible to do in a coherent way for a statistical model, whereas a machine learning model just says, you know, throw all the data in here you can, and I'll give you an answer. And so if we want an integrative taxonomy that incorporates all of these different axes of variation, we need a more flexible computational architecture that can account for multiple dimensions, sort of account for the extended phenotype, if you want to take the Richard Dawkins perspective, to have everything from geography, ecology, behavior, phenotype, et cetera, all together from all of these different axes of organismal variation that are linked to a single outcome. So we need to be able to integrate all these data and get a single set of answers. And I think, or, you know, what I'm proposing is that as we increase the amount of data, even if we're using machine learning algorithms that aren't linked by a unifying model, but more by estimating just huge numbers of parameters, I think what we're going to see is convergence on similar answers. Because if species are individuals, they are phylogenetic lineages that exist spatiotemporally, the more data sets we have, the more data we throw at the problem, the more parameters we estimate, the more we should converge on a single set of answers, because hypothetically, there's only one real set of phylogenetic lineages that has existed in the past or exists now. So what I think what I'm hoping that we're going to see is that as we increase our computational sophistication and our data complexity, we might actually simplify the outcomes as we converge on an estimate of, you know, what are the actual true entities in nature that we're trying to delimit. And so the specific algorithm that I published last year for doing this is a type of neural network called a self-organizing map. I'll just go over it very briefly, but we can have all these input layers. And as I said, they can be traits, they can be DNA, they can be climatic data, and they're projected on a 2D grid. And so those grids are output neurons. We don't have to worry about that, but just the idea we're projecting this into two-dimensional space, and every individual gets projected into one of these output cells in that two-dimensional space. So we can have as many dimensions as we want. We could have 1,000 dimensions, and we can summarize the variation in those dimensions into a single two-dimensional space where things will cluster together in different regions of that space. And so again, it's a fully connected neural network, which just means every input layer is connected to every neuron. So there's lots of parameters. Every single line here you see is a parameter. But what we can do is train this with our data and then get a visualization of how the clusters exist in our empirical data set across all of these dimensions. So just imagine, say, this three-dimensional space, and there can be many more dimensions, but you know, our brains are limited to mostly three. And along all these dimensions, we will see these clusters forming, and we could just say these are two species, and they cluster along these three dimensions. If we project that down into two dimensions, we can pretty clearly see these two different clusters here. And so what a self-organizing map does at a very high level is it takes each neuron and it starts assigning each neuron to a data point, and then warps that output lattice structure to the data points in a way that accounts for the greatest amount of variation. And so if we think about that in multiple dimensions, we can see here, this is our output lattice, and it's warping itself as it moves closer to each of the data points to give the best fit approximation of assigning each data point to an output cell. And the iterations here is, it just does this over and over again. This is the training cycle, the learning cycle, as it attempts to learn the best possible dimensionality of the data. And so over time, if we measure goodness of fit using something like stress or energy or something like that, it will plateau once it's learned the most optimal configuration of assigning those data points to the output. And what that yields is something like this. This is our output. It's 8 by 8, 64 cells. I'll just back up here. So again, we had our input data. We're projecting it onto this two-dimensional lattice. This is our two-dimensional lattice. And every data point each one of these green or yellow data points is being assigned to one of these 64 cells. And so the larger the square, the more data points have been assigned to those cells. These cells are empty. We can also then ask in the original multidimensional space, how close are those cells to each other? So the blue regions, the cells are very similar. The purple regions, the cells are very different. And you can see it's this arc here of these cells that don't have any occupancy that are also very different from each other. What is that saying? It's saying that basically we're clustered into two distinct groups. And we're clustered into two distinct groups based on variation in the input layers of two different axes. In this case, red and green dominate this cluster and blue dominates this cluster. But it could also be, tail length dominates this cluster and chest width dominates this cluster as a morphological trait. And so with this output, we've condensed, in this case, 3 dimensions, but it could be hundreds or thousands of dimensions into a 2-dimensional output with an idea of which of those, the input variables, is actually giving us the greatest difference between the clusters. And the clusters themselves, we can then take to represent, say, hypothetical or lineages or candidate species that we can then test further. So I did this initially with only a single layer of genetic data in 2023. And I've implemented this in a package I'm calling Delimitation. And this was for a salamander from the eastern US called Desmognathus monticola. And I realized when I was putting together this talk, I guess probably a lot of you have never actually seen a salamander. They're pretty cool. They're very common here in the eastern US, and they're one of the main organisms that I work on. And so what this allows us to do is delimit species from a variety of different data sets, so get an integrative taxonomy. And then also at the end, I'll mention potential conservation uses. So this was just based originally on genetic data before I was able to expand it to incorporate multiple axes, including traits and climatic data and spatial data. So this is Desmognathus monticola. This is the seal salamander. And we generated a nuclear data set, nuclear DNA from SNPs using genotype by sequencing. We measured 17 morphological traits. We have all the spatial data in terms of the latitude, the longitude, and the elevation. And I also extracted climatic data from the major axes of bioclim and envirom to climatic data sets that had the most importance for building ecological niche models. So the sort of environmental factors that are the greatest determinants of where these populations are found. And we can train each of those layers individually so that self-organizing maps allow us to learn the patterns of variation within each layer separately, independently, and then combine them in a linked output to a single set of overall output cells that can be delimited as species, while also giving us the importance of each layer, and so telling us which variables are most effective in differentiating between the candidate species. And so it's not surprising that the genetic data had by far the greatest importance, but second was actually the climatic or environmental data, then the morphological data, and the least impact was the spatial variation. So, we can make an immediate hypothesis that there is not a lot of isolation by distance occurring. And we actually tested that using classical models for IBD and found it to be the case, whereas there's a huge amount of ecological differentiation that is reflected in the genetic differentiation. We do some basic clustering analyses. This is just how many clusters are supported by the data. The answer is 2. So it's dividing the original, set of populations that we evaluated into 2 distinct clusters. And this is our output grid. So we've already seen this a couple of times before. And here are the 2 different clusters. So there's a set of individuals that are mapped to cluster 1, a set of individuals, much larger set, that are mapped to cluster 2. And then these are the distances between the grid cells that allow us to statistically distinguish cluster 1 from cluster 2. And then we can map that again, and we see basically the division from northeast to southwest of the 2 populations that we can then treat as species. So this is the southeastern US. If you can imagine, it's North America, like Washington, D.C., where I live, is up here off to the right. This is the state of Georgia. If you've heard of the city of Atlanta, that's right here. And these are the Appalachian Mountains. So this is where most of the salamander diversity in the world is concentrated. And then these are lowland populations. So that's our division here, really. We're talking about a highland species and a lowland species. We can also see which variables are most important in differentiating between those species. And again, not surprisingly, it's primarily genetic data, but we also see climatically that the number of months of high temperatures are very important. Elevation is less important than latitude or longitude. And then in our trait data set, things like toe length and head length, or sorry, humerus length, so the length of the limbs, are a lot more important than things like snout gular length. And so that's just the minutia if you're interested in the species themselves. But what we can learn, from this machine learning algorithm is which of those variables have the greatest signal of differentiation of these lineages, allowing us to then create further hypotheses and test them in more detail as to how they might be related to the speciation process. And so I've made this available. You can download this, and I've tested it across a variety of data sets, so it should be pretty easy to implement and use for almost any context. And it makes all these nice figures for publication where you can input any combination of those data. You don't have to have all four. You can use just genetic data, or genetics and traits, or genetics and space, et cetera, to ask, from a set of populations, a set of individuals, how many clusters do I have? What parts of my data set contribute to those clusters? And which variables within those layers are most impactful for differentiating between those clusters? And so this is really the first algorithm that, it's the first algorithm that I'm aware of that actually allows us to do integrative taxonomy in this context of simultaneously, quantitatively analyzing more than just genetic data or more than just trait data in a single analysis. One thing I didn't even mention, but you might notice, these are pie charts, and they represent ancestry proportions. So you can actually estimate hybridization and genetic admixture from this as well. I didn't go into that just for the sake of time, but you can, if you're familiar with structure or programs like that, you actually get equivalent values for structure plots for estimating individual ancestry from this as well. And so this allowed us to define and describe this new species, Desmogonnathus chiha, from the highlands in the state of Alabama. There are some lower mountains there where this species is endemic. But even in the context of, or sorry, let me back up. As I said before, we want to make sure we're not overfitting these models, that we're not, getting false positives for these taxa, these clusters that we're identifying. And when I was working on the description, I actually found a data set from the early 2000s where a behavioral researcher had actually tested mating behavior between the populations of these salamanders. And they had been treating them all as a single species. And so almost all the northern populations, mated with each other very freely, as you would expect within a taxon. And then they had one population from Mount Chiha, and it refused to mate with the other northern populations. And in the paper, they said, oh, that's funny because they're all the same species. Why would one population of the same species refuse to mate with each other? Because they're all the same species. And then now, actually, we have empirical evidence that these are actually reproductively isolated. They don't mate with each other. So we actually have biological data, even under the biological species concept, that this is a real taxon. It's actually been tested and is sexually incompatible or ethologically isolated, the southern populations from the northern populations. They don't even mate with each other in captivity. So we actually have a classical, biological mechanism hypothesis test that is supporting the outcomes that we get from the sort of, model-free complex machine learning algorithms. And so what this allows us to do, again, just to summarize, is incorporate multiple data layers and data types into what we can think of as being more integrative taxonomy that account for all of these axes of organismal diversity. The estimates themselves are model-free, so we're not getting parameters that we can interpret in a biological context from the model itself. But as I just mentioned, again, since we're scientists, we have the scientific method, we have these biological processes and models in mind, we can then set up further hypothesis tests to sort of rigorously and critically evaluate the hypotheses that we get from machine learning models. And so those are sort of the pros and the cons. And it's also been used, there's a preprint where they use this in Australia and, found a new monitor species, Varanus. And again, the molecular data predominate, but they found that the phenotypic data were the second most important in contrast to the salamanders. And so the last thing I want to mention too is that we can keep adding layers to this. And so one thing I talked about in the paper, but I haven't implemented yet, is the idea that we could potentially implement conservation layers. So we could use machine learning, we could go beyond delimiting species and think about delimiting potentially evolutionarily significant units. So if we have a set of populations, we could incorporate additional layers, things like habitat encroachment or population, changes in population trends, amounts of pollution, amounts of global land cover alteration, things like that, as a fifth layer, where combining that with the genetic data, things like encroachment and habitat loss, might allow us to identify population segments, genetic lineages that are also associated with significant imperilment or conservation concerns from things like habitat loss or human encroachment, where this can then be used as a management basis for identifying areas where spatial imperilment might be higher. And so this approach is not the machine learning approach, but the idea of ESUs is used widely, especially with things like fisheries. So this is the northwestern US, the range of basically the coho salmon. And they're typically broken up by things like river drainages. So, the wildlife officers out there are tending to evaluate things like fisheries by river drainage. Whereas what we might be able to do is incorporate additional layers, as I said, of encroachment or habitat loss or pollution or, climate change, things like that, that might identify different spatial conservation priorities based on the intersection of genetic diversity within populations, where that's interacting with other dimensions of spatial imperilment. So I haven't done that yet. It's something I'm working on now. I'm actually on sabbatical this year, and so I'm hoping to get that implemented in the package as one of my primary goals for the sabbatical. So it looks like I've hit about 45 minutes here, and that's covers most of what I wanted to talk about initially. So with that, I'd just like to thank, Kelsey and the Society for having me here. A bunch of people that helped me out developing these algorithms, as well as GW, our computing resources that allowed me to train and implement a lot of these models. And then NSF provided a lot of the funding for the initial rounds of this. And so with that, I'd be happy to take any questions and be interested to see what you guys think about all this.
Awesome. Thanks so much, Alex. I'm going to ask folks from the floor if they have questions first, because otherwise I would tend to dominate the question and answer period. So folks, if you have some questions, you can either type it into the chat or you can unmute yourself and ask.
I think you can start with your questions and we'll see
Okay, great.
how they come.
So Alex, that was really cool. Thanks so much. I'm quite excited about this at first I was a little wary of like machine learning and systematics because I mean, yeah, I'm from GW, so and one of the things that I found really interesting in how you're pitching this is it's generating subsequent testable hypotheses. So it's not like the tree is the end goal here, but it's rather these groups that then we can learn more about how like speciation processes work. And so that's just more of a comment than a question to start off, because I thought that was a really cool perspective. And then the question I actually have is about the genetic data. And I wondered if the reason it dominates in how it is in your data set is because it's more abundant than everything else.
So yeah, that's a really good question, and I have a whole section in the original paper about that. The answer is no, surprisingly. So the way the self-organizing map, we call them SOMs, the way the SOM package is set up, I didn't write that. That was written decades ago by some other researchers. They implemented a penalty, not a penalty per se, but a weight. So there are weights on the layers by size. And that is intended to equalize the contribution. And there's no guarantee necessarily that that would not result again in overwhelming dominance from the genetic data. So what I did was I did a bunch of simulations. And so I simulated genetic data sets of the same size and combined them with trait data that had zero signal or had full signal. And you actually get the delimitation can be driven entirely by the other layers, even in the face of a large genetic layer when the large genetic layer does not have strong signal in it. So if you just generate a random genetic layer, it will support one cluster. So it doesn't automatically delimit clusters when you have large genetic data sets, which is one of the primary criticisms of a lot of species delimitation algorithms, is that any genetic structure that they have will always be identified because the algorithm is designed to find structure, so it will always do that. That doesn't happen here. And if you remove the signal from the genetic layer, there is still signal for two clusters in the morphological, spatial, and climatic data. And so I ran analyses that had huge genetic layers with random data in them in combination with the actual real morphological, environmental, and spatial data. And that still results in the estimation of those two clusters just based on the impact of the other layers. So the size of the layer, at least with the sort of basic tests or preliminary simulations I did, doesn't seem to be driving it. You can get signal from the smaller layers when it's present.
Oh, that's good. That makes me feel a little better, because at first I was like, well, clearly, like, this has to be driven just by like your massive GBS data set. But so then something else I was wondering about is like, would you be able to quantify like variance among the different groups, like how variable each of the clusters are relative to each other, or is that not really? Like, I think
Potentially,
about variation, right?
potentially, yes. I haven't. I've tried to do that in one sense, but not, I think, the sense that you mean. So when you have you run the SOMs hundreds or thousands of times, and each time you do that, the assignment of the clusters actually changes between the individuals sometimes. So when you have individuals that are on the edge of clustering, they can switch back and forth. That's where we're actually estimating the species coefficients, like the potential hybrid ancestry. So you do get an estimate of which individuals are always fixed in species A and species B, and you also get an estimate of which ones are potentially switching back and forth and are less fixed in their classification based on the variability in their data.
Oh, that's cool.
If you're thinking about variability within the clusters with respect to the input layers, I haven't actually done that, although I think it would be relatively easy to implement. So it wouldn't be hard to come up with some estimates of that. I hadn't actually thought of that yet.
Oh, well, please get on that.
So yeah, I'll put it on the list.
Awesome. Thanks. Are there any other questions or?
I haven't seen any at the moment. I
Okay.
just wanted to say that it's quite interesting, and I think, from a bacterial perspective, we have lots of sequence data and genomes and things, and it would be interesting to do some of this. Unfortunately, phenotype is not our strong point. They vary quite a lot, but it would be interesting. I think for the mycologists, we also have some mycologists here where you have morphological data that could also be quite interesting when you start combining that.
For sure. For sure.
No, I would love to see some like stress testing cases, where you're really pushing the boundaries of what we might consider phenotype or ecology with things like bacteria or fungi, where the dimensions that you're actually measuring are not just things like arm length or chest width, but more subtle aspects of environmental variation. And I'm not sure how it's going to behave in those instances, but I'd be very interested to see.
Lichen. You should think about lichen.
Well, I mean, I think that's what you might actually get, is that if you applied this to something like a big bacterial data set or a fungal data set, it might actually start delimiting like meta communities instead. So you might start seeing like networks of related taxa or related genotypes that like co-occur environmentally, which probably isn't the same thing as actually delimiting species, but it might actually unlock a sort of a different avenue of investigation in terms of finding associations between like those sort of meta community dynamics.
Yeah, for sure. Yeah. I was at the risk of like just rambling because this is quite exciting to me, but I was thinking about like microbes in like plant communities and how they're helping with establishment, and we don't like learning more about what microbes are available are there and how those communities change across different taxa. This would be quite interesting, but I digress. So
We have one question here from Hanley. Let me just get to the chat. It says, can you briefly explain how the sequence data is converted to a matrix when running the analysis?
Yeah, that's a good question. It's kind of, if you're familiar with R, it's an open source computing language, and it's extremely flexible and powerful. And so I struggled a little bit when I was building the package because there are a million different ways you can do it. And so I didn't want to leave it down to just say like, oh, do it however you figure it out on your own, because that would be too open-ended. So I have it set up to use a specific package called a dgenet and a specific type of data object called a genend object. But, any way that you load in the data into R and convert to a matrix could theoretically be used and probably get the same results. I think the best way to do it, though, is, especially for something like SNPs, you typically just have a SNP matrix, from your sequencing run, where it'll be a 0 or a 1. Do you have allele A or allele B at that SNP? And if you have 0 and 1 or 0.5, if you're a heterozygote, you can actually just load that directly into R and really just type, as.matrix DNA or whatever you've called the object, and it'll automatically convert it. If you have it in a sequence format like phylip or FASTA or NUIC or something like that, you will have to convert it somehow. And there's a bunch of different routines that are available in R that can actually take A, T, C, and G and convert it to 0 and 1. And so I have in the documentation, my preferred pipeline, as I mentioned, for a dgenet and genend objects, where it's just really 2 or 3 commands. So you can load in a FASTA file, you load it in as a genend object, and then you convert the genend object to a matrix. And so I have the commands for that. It's the funny about machine learning is there's no right answer, there's no wrong answer. You could literally put anything in and it'll give you some kind of output. It's just a question of being rigorous enough to make sure you haven't made some mistake along the way.
Thank you. Hanley saying awesome, thanks.
Yeah, no
Any
problem.
other questions that somebody would like to ask? Here's Hendrik Newman say, really cool talk. Would it be fair to suggest that based on your work, a modern systematic study would be perfectly fine on its own, and the results from your pipeline can then be used to delimit morphologically, etc.?
Yeah, I think that would be a perfectly reasonable response. I mean, especially when we're dealing with unknown taxa or things that are difficult like bacteria or like microbes or fungi, or even amphibians and reptiles where we just have no idea. We could go out into like the Congo Basin or something and collect 1,000 frogs and we'd have no idea what they are. That's the power of 200 or 300 years of classical systematics that we have, is the ability to make those sort of first pass judgments, to sort of define morphological species groups, et cetera. And then something like machine learning species delimitation can just form a complement to that, where something like DelimSom works best when you already have sort of a rough idea of what the structure in your group is. And so having, classical systematic studies would be a great preliminary or prelude to that, like an a priori set of analyses that then allow you to sort of focus your questions, get your population sampled in the way that you want them, sample the traits or whatever data layers that you want to have, and then use that either in a validation sense of, validating the previously hypothesized species, or generating candidate species that you can then test as hypotheses with like additional fieldwork or something like that. So either way you wanted to do it, I think would probably be valid, philosophically, scientifically, etc.
Okay, thank
Awesome.
you.
Awesome. Sorry, I just wanted to just correct the way I was phrasing that question. It was mostly just in regards to how powerful the signal is for the molecular data. It kind of gives credence to the approaches where people kind of like to blindly go into a space, basically collect as many taxa as possible, and then try to delimit species from there for specific groups. It seems like your data suggests that with a machine learning approach, there could be a very strong, unbiased starting point for addressing like the taxonomic impediment that we have for the amount of time that it takes to actually delimit these species just based on morphology alone. This would be a very fast way to kind of in an unbiased approach, just give us a very initial idea of how many taxa there actually are, and then with basically like this molecular data, or at least with these clusters that your pipeline would throw out, actually then use that to delimit the species, do the taxonomic work thereafter. So that's actually just such cool work that you guys have done. So it's just really, really awesome.
Oh, thank you. And I think you characterized it pretty accurately. I think that would be a good way to do it. I mean, you can make a first pass, you can crunch a ton of data initially down into a set of initial candidate species, and then use that as your starting point for more in-depth or detailed assessment of things like morphological or phenotypic data. And then that's sort of the inverse of, I think, Kelsey's question, which is that I also did some simulations to show that even with morphological data sets alone without molecular data, you can also generate robust estimates of clustering as well. So even if you have limited or sparse molecular data, you still have the potential for using these algorithms.
Absolutely. It's just a very awesome starting point when you have so many species to describe and so few actual taxonomists to do the work. Right. If you can actually just start off with here's places that you can actually focus on, maybe give them a very good starting point from which to then delimit species, and maybe even give a rough estimate for specific areas, like you said with your work, specific areas that are important for conservation before we even know how many species there are. This preliminary data could be used to suggest there's quite a bit of variability and this specific spaces need to be protected before we have even been able to describe all the species there, and then also maybe highlight specific spaces that need extra work, whereby there are these hotspots that might need a bit more work done. That would be a very cool way of like identifying those.
That's my hope. That's what I'm hoping people will take this and run with it and try to do that kind of thing.
Okay, awesome. Thank you so much.
Yeah, no problem.
Any other last questions that somebody would like to ask? Doesn't sound like that. So thank you, Alex, for your time, and for everybody for joining us today, and for a really inspiring and interesting talk. I think this is a little bit of a view to the future, what is coming, and as you say, you've already seen the writing, and I hope some of us are now also start reading what's on the wall, and would like to try some of those things. So thank you very much for your time, and thanks for Kelsey for organizing this.
So thank you for having me. It was great to be here.
Great.
Have a great day.
Oh, you too. Have a good one, guys.
Thanks,
So thanks for everybody for joining us, and we will let you know about our next one, which is probably towards the end of the year. Bye.
Bye. Thanks, Venice.